All right. So hello everyone and welcome to this session about Introduction to Bioinformatics. So the OmicsLogic Introduction to Bioinformatics program is an introductory program in the field of bioinformatics, or you can call it the intersection of bioinformatics and biology. In this program, we will discuss the changing landscape of data-driven research in life sciences and the opportunities that comes with widely accessible high-throughput technology and computational methods for analysis and annotation and detailed data research in industry. So we will be discussing about various data types and emerging technologies and their impact on health and healthcare, agriculture, environmental sciences, etc. So today, let's learn more about the program and also data-driven bioinformatics. So why we are doing this? So one of the reasons is that because biology is rapidly acquiring the character of data science, billions of data points on genes, proteins, and other molecules are compiled in those large files, and they have been systematically studied. So one of the reasons is to be able to analyzing this kind of data that will lead to more knowledge and understanding about living organisms, including human health, biotechnology, crops, and livestock. So course will this course will give you an overview of all those techniques, and this program will be guided by highly experienced mentors with degree in bioinformatics and computational biology. So my name is Mohit Mazumdar, and I have done my PhD in bioinformatics and structural biology, and in Fine Biotech, I've been working and mentoring students for the application in their research project and machine learning for collaborative bioinformatics projects. And along with me, I have my colleague here, Harpreet Kaur, and I'll be introducing her later as well. But she's an expert in molecular biology and biochemistry, but because she did extensive work in her master's and in her graduation, finishing those, learning about those basic areas and techniques, building upon the foundation. And then she went on to do her specialization in cancer genomics, where her research was focused on genetic biomarkers for the diagnostics and prognosis of liver cancer. And during that time, she was able to publish a lot of articles. So what I'm trying to say is that this program is supported by a lot of research, people with a lot of research background that helps, and the reason is to be able to help participants to understand the bioinformatics, the application of bioinformatics in research, and especially at this time, at this modern biology, in this era of modern biology and data-driven biology. So during this meeting, as Deeksha said earlier, we will pause in between to address your questions that you put in the chat box. So there is a chat box as you all have access to, and you can ask questions there. Also, as Deeksha mentioned, and all of you already have, so to keep your microphones muted, and we will have a Q&A session towards.
Mohit, you're muted. Mohit, you are muted. We cannot hear you.
So I guess I was muted back there. So as I was saying that this online program will commence from 13th October 2020 and will cover some of the important technologies and its application in today's world, as I was telling you before. So the participants of this program will be able to access bioinformatics resources along with expert and mentor support to get the help with the entire learning process that will be available for of biology or to data driven biology. So preparing biologists and clinicians, especially students and people who are preparing right now, people who are doing research right now, people who are involved in any domain of life sciences and are working on some sort of biological problem, or students who are learning this very concepts of biology. So one of the reasons is that preparing biologists and clinicians for the influx of this data that we will be talking about, this big data, associated methodologies, and data-driven research projects. So these are some of the objectives. So data-driven projects, because it's prominent as emergence of computer for tech industry. So an importance of doing a project is because it gives you an experience that you gain out of it by doing a research, by doing an independent research where you're trying to address a unique or a novel problem. So that holds a lot of value. So as I was telling you about the program, about the OmicsLogic program, uh, we will be combining several different components. There will be several different components, and we will be combining those components for this immersive experience. So in Pine Biotech, we have been doing the OmicsLogic programs for a while now, and been collaborating with several universities. So in this experience, in this research experience, we will combine, as you see, online interactive sessions like this, where we will be learning about the courses, learning about the self-guided material along with the session. So once this session is done, as we will be doing in the program, it will be followed up with assignments and courses that are relevant to the session, and that will enhance and give you a deeper understanding of what we are doing and what we are trying to achieve. So one of the reasons of having this is to be able to go into depth in much more details, because research takes time, as you know. So these associated materials help a lot to gain such objectives. So again, the third aspect, the very, very important aspect of this experience is this unique experience, I must say, because of this Bioinfo platform and also the OmicsLogic platform, which we will be learning about in a few more minutes. But to tell you more, it gives you a practical hands-on experience working on real data sets, and to be able to implement your ideas on that real data set. So we will be learning more about it in this entire program. We will be addressing research problems. We will be looking at the problems that you are proposing. So it will be an immersing experience, I must say. So to give you an overview of this introduction to bioinformatics program, this program is built to give you an overview of the next generation sequencing techniques that have evolved with this high throughput data, and also the different application of this technology that has transformed how we look into different biological problems related to healthcare, related to translational bioinformatics, which is being applicable to look at mechanistic details, or how entire cell is being modeled. So introduction to bioinformatics gives you an overview of that. So yeah. Introduction to Genomics is again a free course. So these free courses are available for everyone to take and to learn about the basics and to get an idea of what it will be in the program, which will be more elaborative, going within all of these technologies, some of these technologies, and learn in depth. So as I've been talking to you about the characteristics of big data, I mean, it's really very easy to say and really difficult to visualize because it's not a physical thing that you can see, but to understand that what it can, sort of the components of what it can be is it depends on the velocity, veracity, and variety of this data. So we will be discussing about those, and those will be covered and are well described in the courses. So you will find videos and you will find different other glossaries and quizzes to be able to understand that, to be able to judge how much you have learned. So as a result of this, anyone starting on this journey in bioinformatics can start developing a project of their own by seeing how they are doing, how by seeing their own progress using this platform, which we'll be discussing in more detail later on. So this includes, certification from Pine Biotech and certification from Tower Bioinformatics Research Center, that gives you an experience, and this experience is also a part where where you will be working on project-based methodologies, where we will be discussing about how you can transform theoretical ideas along with the hands-on training and independent thinking that leads to this research experience. So it's a complete transformation, learning the technology itself using the courses and the sessions, and then doing the hands-on along with us, followed by your independent thinking of the application of what you have learned, and then the research experience that you gain from working on this entire program. So these are the major components of this entire program, and the way it works is that we will be going through a publication where we will be showing you the data from that publication and the methodologies, and then we will be using our own skill sets that we have acquired, and then on independent data sets that we will be discussing during the program. So as I was coming to the independent project, so after doing after learning through these lessons and after doing some of the courses, we will be seeing some of the examples from the publications that we will be covering during this course. So those publications are those publications that have data sets associated with them. So those data sets will be used in this program where we will be analyzing those data sets, and we'll be talking more in detail about those data sets in the next few slides. So the entire program encompasses different components, as you saw, from courses to projects. So to give you a lot of examples on different kind of, may that be neuroscience, may that be infectious diseases, may that be cancer biology, may that be biotechnology. So we have several of these examples in the platform that you can take and learn, so that the purpose is to be able to see the examples from these highly, well digested publications, and to be able to implement them for your own independent research. So as I was telling you, the practical aspect of this is the Bioinfo platform. So the Bioinfo platform will be available to the participants, and the second platform that will be available to the participants is the OmicsLogic code platform. So we will be going into looking into the NGS data and its application, and then we will be learning about how to do the backend, how these programs work in the backend, and how we can visualize and manipulate the data based on by using programming or coding language such as R and Python. So we'll be using the OmicsLogic code playground that we have recently started, and this code playground has capability of handling both Python and R code, and it's intuitive, again, a platform built for biologists, so that you can actually learn how every component of the processes that you've been using works at the back end, and learn to change things, and learn to change things for better visualization, or to be able to learn about the data in depth. So that will be a part of it. So as I was telling you that we have been doing this for a while and have had several reviews from participants all around the globe. And we have been working with many of the participants and researchers to be able to translate this learning into a project or into something, a poster or a publication. So let me now go back to this chat and look at the questions that you might have. So I see a lot of okay, Deepsha, thank you for sharing all the links, but I think I would stop here for a while and let anyone, if anyone have a question, can go ahead and ask that question. And so if not, so let me introduce everyone to Harpreet, as I was telling you. So Harpreet, let me pass it on to you, and then we will learn about the program in more depth. Thank you.
Thank you,
Dr.
Dr.
Mohit, we have a question here on the chat.
So I see a question from Duvani. What are the future aspects of bioinformatics? So Duvani, the future aspects of bioinformatics are the different aspects of bioinformatics, or you can say the application of bioinformatics, data-driven biology, as if I may say, are in therapeutics, in pharma, in agriculture, because we are looking at the genomic data, we are looking at the transcriptomics data, we are looking at the epigenetic data, we are looking at the metagenomic data, and there are different other omics technology that have been and that are evolving every day. So it's important to understand how it works. I mean, how do you see from the machine how the machine is being able to generate this data, and then you being able to understand the meaning out of that data. So this entire process, if you are able to understand, if you are able to learn that, so that actually helps you in several ways. Because if you are doing research, and if you are the person who are doing the research and who is actually generating the data, and I think you are the best person then to be able to analyze that data as well. So what we have found is that there is a gap in there in data analysis, and it's nowadays, if you look at the opportunities, a lot of opportunities are in this field of data analysis, and especially big data analysis, like the omics data, as I said, and also clinical data. How clinical data, we are learning about COVID-19, and so much of data has been generated for COVID-19 itself. So why do you think that is happening, and what is that leading to? So all these publications and all this big data that is being generated every day is being analyzed by the experts, and then we are coming to those, all those decisions that are leading to those kind of interventions that we are trying to make. So may that be cancer, may that be any disease, we are generating a lot of data, and that is because there are so many patients. So there are so many patients and there are so many time points. Just to imagine, like one person's genome would take roughly around 200 gigabytes of data. And then if you go on and add to that, like if you take time lapse, and so how much data that would be. So the important thing is to be able to learn about that and to be able to be able to understand that how it actually works. So if you do that, if you were able to learn that, I think it really helps to make those decisions that are really, mm, are translational and helping or to help others, so to say. All right, then. Thank you, everyone. So to bear with me, and let me pass it on to Harpreet. So she'll be taking the next part of the session.
Thank you, Dr. Mohit. I think, okay, let me share my screen. So about me, my name is Harpreet Kaur and I have done my PhD in Bioinformatics from CSIR Institute of Microbial Technology and JNU New Delhi. My doctoral research was focused on the identification of genetic biomarker for the diagnosis and prognosis of liver cancer. Currently, I am OmicsLogic trainer at the Pine Biotech. Now let's move ahead and see what we are going to offer under this program. During the upcoming program on Introduction to the Bioinformatics, we have 6 interactive sessions. Sorry. We have 6 interactive sessions. You will get to see the broad applications of next generation sequencing, biostats, and tools for the big data analysis. In practical assignments, we will learning about the logics of the bioinformatics and see how data science methods can be used to study biology. Eventually, you will also able to plan how to take what you have learned and apply it to an independent research project using tool and the methods that we will cover in the program. Now, let's look at each session one by one. In the first session, that is Introduction to the Bioinformatics, we will be focused on the concepts and the applications of the big data. Here, our platform, you will learn our platform T-Bioinfo, that is an initiative and user-friendly interface for analysis of the big data like NGS genomics, transcriptomics, mass spec proteomics, and metabolomics and structural biology. And you will learn about how you can apply machine learning to analyze phenotypic and visual information. You will find various educational material and projects that overview different topics where you will cover different skill sets. There are various big data resources like NCBI, that is National Library of the Medicine. As we are looking at this graph, at x-axis represented the years and y-axis representing the number of the sequences in the millions. Since the discovery of the structure and the function of the DNA, there is a large number of genomes are sequenced every year. First human genome project was sequenced in the year nearly 2003. That brought a significant number of the whole genome. And then later on followed by in the year 2015, 1,000 genomes were sequenced. And they brought a significant number of the whole genome sequences to the research community. NGS continues to emerge. And by the year 2018, there are nearly 1 lakh whole genomes were sequenced. As a result, lot of data from experiments is available, and today there is ever increasing number of the data sets available that help researchers understand cellular processes. Till date, Gene Expression Omnibus, that is one of the biggest repository at NCBI, alone contains more than 80,000 public data sets that span hundreds of the tissue types and in thousands of the organisms. This program is designed to highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for the precision diagnostics, big data integration, and machine learning, and transforming personalization of treatment selection. And innovative algorithms allow for unprecedented capability with drug discovery, design of large molecules, and drug repurposing. Second session will be dedicated to the introduction to the genomics, that is DNA variants and the mutations. Here, we will learn about the shotgun sequencing reads, the need for the structures data, and associated bioinformatics methods that include processing of germline and somatic mutations, and then analysis like mapping and the detection of the variants. And we will also learn about interpretation of the data, like we will interpret what are significant and insignificant mutations in particular data, and ultimately how you can elucidate a significant biological interpretation from a big data. Next, we will try to understand central dogma of biology, where we will also look into how different codons provide instructions in assembling amino acid to form protein. We will also look into how a point mutation in a codon can alter the functional aspects of proteins. With the help of a Bioinfo platform, that is our server platform, you can visualize what kind of and where is the mutation taking place on a chromosome. For instance, here we can look at the position 743, G is getting mutated to the thymine. So this is how you can visualize what kind of mutations are taking place in a particular gene or a chromosome. Further, our platform offers you to analyze or visualize how your reads are mapped onto a genome, and you can accordingly analyze these reads. Our Bioinfo platform will allow you to analyze genomic data. We will learn how we can perform various types of mutational analysis using Bioinfo platform. On the Bioinfo platform, particularly the genomic data section, offer various types of the pipelines that will help you to understand the different steps that are involved in a mutational analysis. For instance, under the data pre-processing step, you can understand which algorithm need to apply for the pre-processing of the genomic data. Then under the mapping, there are you can understand what kind of the algorithm and why you need to implement particular algorithm to align or the mapping of your reads on a genome. And then using a variant calling, using a variant calling option, you can compute various kind of variant calling from the data. And then differential variant calling option provides various algorithms to compute differential variant calling analysis in different groups of the samples. Then using the annotation option, that will allow you to annotate your analysis, and you can also finally post-process your data using various methods on Bioinfo platform. There are the different courses, that is like Introduction to Genomics and Genomics 1 at our educational platform. where you can find the details on the genomics and genomic data analysis. These courses provide deep understanding on why do we need genomics to study, then what are different logics for the genomic data analysis, then what are different algorithms or tools available to analyze a genomic data. Then beside, there are various kind of quizzes and assignments are on this course that will enhance your understanding regarding genomic data analysis. The next session will be dedicated to the introduction to the transcriptomics or gene expression data analysis. Here, we will learn about how we can measure functional proteins by the way of mRNA abundance and accurate identification of alternative splicing, an overview of the RNA-seq, read quality, pre-processing, mapping on a reference genome using various tactics and logics. And we will also learn about alignment free quantification, and quantification of the genes and the transcripts. Then we will learn about how we can prepare a data set to design any research project. In our program, we will try to analyze breast cancer subtypes using different data sets from different studies like PDX models and cell line data. These breast cancer subtypes are different from each other owing to it's the presence or the absence of various types of receptors on the cell surface of these cell lines. Eventually, they alter the nature of the tumor. That is, either one type of cell subtype is more aggressive, other is less aggressive. For instance, we have data set for the that is ER positive or estrogen receptor and triple negative. In the estrogen receptor positive cell line subtype, cancer is identified by the presence of an estrogen receptor on its cell membrane. While in the triple negative, none of the three common receptors are present on its membrane. This is just a one difference, and as a result, one form is more aggressive and other is more treatable. Besides, there are mice models and like XID, NOD, athymic, and SCID mice. So now in our program, we will try to perform various kinds of NGS analysis using datasets from patient-derived xenograft or PDX model. Now let's understand what is a PDX model or PDX xenograft. PDX models are generated when a fresh tumor tissue obtained directly from the patient is implanted into immune deficient mice like SCID, nude mice, etc., subcutaneously or orthotopically. As such, these PDX models maintain the principal histological, clinical, and molecular characteristics of the original patient tumors, while remain biologically stable itself. Since PDX models are more closely resemble with the and recapitulate human tumor than the standard in vitro cell line and xenograft approaches, they remain a key experimental platform for preclinical drug development. Other study involves the cell line, breast cancer cell line, where nearly 80 cell lines are treated with 138 drugs. The response of these drugs is measured on various types of the omics data. For the training purpose, authors use the cell line data, and for the validation or the testing purpose, authors have used TCGA, that is the cancer genome samples. During this program, we will using these data sets from these well-established studies. This analytical platform, that is Bioinfo platform, will provide hands-on experience for anyone to apply learned skills to interesting data set with color-coded logical maps, explanation of the methods, detailed inputs, output formats, and reproducible workflows. So let me just demonstrate how we can generate RNA expression table from the RNA-seq data using this simple Bioinfo platform without having any without having any coding knowledge with you. Now let me go to our server. So this is our T1 platform that is available at server.t1bio.info. So here I am just giving a one demo how you can generate a RNA expression table from a raw RNA-seq data. So I'm here using demo RNA-seq PDX project. Once you click on that project, you will find a demo pipeline here. Once you start, before starting, you must have otherwise have data, as this is a demo pipeline. So we already have input data here. So let me click on start. Okay. So it will first on clicking on the start, it will provide you details about these data, about these each step. And under the input pipe files, we have already uploaded various files, like already uploaded the reference genome as well as the files that we want to analyze. So once click on, so the next step is pre-processing, which is indicated by this blue color. So it includes all the pre-processing steps. So first of all, thermometric step. What it does, it removes the adapter from the reads. And detail of each step, whenever you click on the step, you will find details regarding each step and why we are doing, and all these steps are, you can even find details here. So once I click on that, okay, so my thermometric step already performed. So let me go to the next step, that is PCR clean. PCR clean, what it does, it removes duplicates from duplicate reads from the data. Then the next step, that is represented in the red color, it's showing that next step, that is mapping or the alignment of the reads on the reference genome. You will have various options. So depending upon your analysis or what kind of data you have, you can choose an option and algorithm. So I'm here choosing type T2 as it will generate the transcript. After mapping of the reads on a genome, then we can generate a gene expression table using RSM expression table. So here you can generate read counts as well, FPKM, TPM, etc. So that's depending upon your analysis or your requirement in your project, you can choose an option. So by default, it's choosing FPKM values. Then we can also perform differential gene expression analysis. So here I only want to generate a gene expression table. So I'm skipping the differential expression analysis. So I am just going to end my pipeline here. So once you end and run your pipeline, you will find your output files under the option download pipelines output files. Here we will find various kinds of files. So I am here checking this RNA expression genes with FPKM text. So after running this pipeline, we have obtained a well-designed table where samples are present in the column while genes are present in the row. So this is how we can generate or we can perform a complex analysis using a simple T-Bioinfo platform without even the knowledge of any programming language. So this is how we can perform different kind of analysis using T-Bioinfo platform. Now let's move again back to the presentation. So this is T-Bioinfo platform where you can perform various kind of analysis on your data using based on your study design. Okay. The next session will be dedicated to the BioML session. That is includes statistical analysis and machine learning on our data. Here we will learn about the statistical and ML approaches for analysis of biomedical data using our T-Bioinfo platform. We will go through statistical approaches to study processing pipeline outputs, perform exploratory data analysis, hypothesis testing, data mining, and classification methods. We will also understand different methods on comparative data analysis, association visualization of complex and high dimensional data using a very simple platform, that is T-Bioinfo platform. So let's how we can perform various kind of statistical and machine learning analysis. Let's say we collected the data on eye color, gender, age, height, and weight of the people. We hypothesize that some of variables are related while others are not. We also think there are exceptions to this hypothesis. So what could be some are related, like height or the weight are related, but how much they are related. We can analyze what kind of factors that are related or what kind of factors are not related, and if they are related, how much they are related, or how many factors are interlinked to each other. So we can learn about these kind of analysis using various kind of statistical and machine learning approaches. For example, here weight and the height are related. So if we plot a trend line using height and the weight, and we have observed some of the dots are close to this line and some are far away. So is there any way to quantify this relationship that we found? So you can find answer to these questions implementing various statistical and machine learning approaches. So on our educational platform, there are 3 courses on transcriptomics, like transcriptomics 1, 2, and 3, where you can find the details on RNA-seq data analysis, pre-processing, and various kind of statistical and machine learning analysis and methods. These courses provide deep understanding on NGS RNA-seq data, pre-processing and normalization of gene expression data, then how to implement statistical and machine learning approaches or methods to analyze transcriptomics data. And you will also get various different assignments and quizzes that will ultimately enhance your knowledge and understanding regarding gene expression data. Next session will be dedicated to the NGS data analysis in R and Python. Here we will learn about methods on NGS comparative data analysis, association and visualization of complex and high dimensional data using T-Bioinfo platform and R and Python language. Coding challenges and problems that get you motivated to think outside the box and learn about the biological phenomena as you gain coding skills. This is our code OmicsLogic platform. So once you have learned about and understand about what kind of steps and what kind of analysis you can perform, you will explore the coding behind each step, and so that you can understand how it works and modify it to ultimately to fit your project and research design. Our code omics Logic platform has various tutorials that will help you to learn and understand the coding in the R and the Python if you're willing to learn the coding skills. So once you have learned about the concepts of the bioinformatics, NGS, next you can proceed to design your own project. You can get various types of data from research projects or various types of repositories like NCBI Geo, SRA. Then there is also other repositories like on cancer, it is the cancer TCGA, that is the Cancer Genome Atlas. So then you can plan your project and project proposal, hypothesize what kind of study you want to do. Our team will help you during your research project design and analysis. So finally, you can learn and present your scientific data at conferences or at the public, or you can present in the form of publications. This introductory course leads into multiple courses and projects that can serve as a springboard into the world of the bioinformatics by seeing how experts analyze their data and achieve various research results. You can start developing research question of your own and identify the appropriate type of data at different repositories and plan your research problem accordingly. So this 1-month bioinformatics training program will include several components already Mohit had defined and we have covered so far. So this is how you can proceed. So if you have any questions, I think already you have a lot of questions. So just let me look at the chat, what questions you have.
Dr. Harpreet, I've filled in everyone with the questions that they have for now. If anyone has any other questions, please let us know.
Okay. So if there is no more questions, let me pass it to, okay, I think that's one question. Let me pass it to Ms. Beepsa, who is our community manager, and she will be able to guide you with the program registration and access to various resources that we have discussed so far today. So over to you, Beepsa.
Thank you, Dr. Harpreet. I'll start sharing my screen.
Okay, just stop.
I hope everyone can see my screen right now. Okay. So I wanted to walk you all through the resources of this training and also the registration process. So this is the registration link to this program, which is the bioinformatics.omicslogic.com. So once you come up to this site, you will see, once you scroll down, there is a program overview. There is a video recorded, which you can watch, which explains what the session is about. And you will be able to see the syllabus here, which Dr. Harpreet explained. And you can see some of these resources mentioned. So how do you proceed with your registration? So you can click either on the pricing tab or you can click on get started. It will both come up here. And you will see that we have 3 levels of membership options that you can choose from. One is a beginner, another is an intermediate, and the other one is an advanced membership. So I will quickly explain what are these membership options. So the beginner membership covers all of the things which we have mentioned until now. All of the resources you will have access to for this 1-month period starting from 13th October. The only difference between these 3 memberships is in this option, which is the mentor support and an extra time for research projects. So the intermediate membership allows you one-on-one mentor support with one of our mentors. So for example, if you are having some doubts or questions, or if you cannot understand a pipeline, then you are eligible for a one-to-one session over a Zoom call, just you and the mentor, who will walk you through any of your questions that you have. So that is what the intermediate membership is about. While the advanced membership gives you the chance to not only work with a mentor on a one-on-one session, but also the chance to work on your own research project for an additional 30 days. So this is what we are making it available for anyone who is working on their own project. So if you choose this option, you can actually work on your own project, not for 1 month, but for a 2-month period. And you will also have the chance to interact with our team of interns who are involved in other projects of their own, and you will be able to get the guidance of our team. So that is what the 3 memberships are based on. But if you are just looking to learn something and finish a program, you can just choose the basic membership. So once you select the basic membership, you see there is a card option here. You can just use this option, and we are having a code right now. It's called Intro20. So I'll also post the code right here, and I'll also email everybody later on. So once you apply the code, it gives you 20% off, which is just for $56, this program. And this code is applicable for all the 3 memberships running on till October 5th. So anybody who wants to avail this opportunity, you are welcome to avail this opportunity. So what happens once you finish the registration process? So you finish this process. So the next step is you will receive a payment receipt and the details to go into this page. So this is what we call a forum page. It's a page, or you can treat it as a classroom for this program. So we have, for all of our OmicsLogic programs, a dedicated classroom where people are learning from. So this classroom comprises of these sections called the forum, courses, projects, progress. So this is where you will be able to come back and see the entire program and go through the entire program. So I'll be taking you through a few of these headers right now and see what they do. So for communicating with everyone on a day-to-day basis within the program, this is not just about the mentor support, but also about just a day-to-day basis. If you have a problem, we solve it here. So you see there are 2 topics right now. So we create several different topics. It's very easy to create a topic. You write here a title, you post something, and you click on submit. So you can post any questions that you have regarding the sessions, or if you need any help, you can just post it here, and we'll be reaching out to you within the duration of the program. Next is, where do you do the asynchronous courses that we have been talking about, both Dr. Mohit and Dr. Harpreet showed you. So the asynchronous courses are right here. So you'll be able to access all of these courses which are mentioned here. So as you know, Dr. Harpreet mentioned transcriptomics 1 and 2 for learning about RNA-seq analysis. You have introduction to bioinformatics, introduction to genomics, and so on. So you will be able to access all of these courses within this 1-month period. And for someone who was asking about the course PDFs, yes, you can download each of the course PDFs from these courses, and you can save them. So moving on, another thing that you would be able to see are also projects. There are a few projects that we make available. Like right now, Dr. Harpreet was talking about the PDX project. So there are a few projects which are added here. So these are like the asynchronous courses, and you will be able to do these projects. So these would be serving as an example to how you can work on your own research project. And you will have the syllabus right here. So how do you get to the syllabus tab? If you click on the menu here, the syllabus opens up. So you'll be able to see the different dates for the programs and the topics that are going to be covered. And the next thing that I wanted to highlight is a profile. Each participant gets their own profile. So Cindy, whose example I'm taking, is an intern with us, and you can see that she has a completed profile. By a completed profile, I mean a profile photo and her full name. She has also added her LinkedIn ID and her ORCID ID. If you are a researcher and you have, you want to make connections beyond the program. A program is not just like for a month and you get a certificate and you forget about it. You can actually make lifelong connections. You can collaborate with people. So that is exactly what you will be able to do via this profile. And you see her membership level is stated that she is in an internship. She has earned points. And coming back to points, I just wanted to point out to everybody, we give out two different certificates. One is a certificate of excellence and a certificate of completion. So anybody who has over 10,000 points in a program receives the certificate of excellence. And the way you earn points is by doing these different courses, running pipelines, and going through the different tutorials and lessons on the Code OmicsLogic platform. So everything is integrated. It is everything you can see here on the activity tab. You will be able to see not only your own progress, you can also go to anyone else's profile and see theirs as well. So we'll be talking more about that during the program on how you can see progress and how you can collaborate with each other. So you can see what she has done during the entire length of the program. We can see here. And moving on, I wanted to invite everybody to join our two groups. So we have this one group on LinkedIn called the OmicsLogic Machine Learning, Biomedical Data Mining, and High Throughput Data Analysis that you can join via here. And we keep sharing resources, details, and also any job opportunities that are available in this group in LinkedIn, so you can connect with us. And we also have this one group in Facebook, which you can again join. So please check them out. And I also got a few questions from others who mentioned that they are already familiar with the basics of bioinformatics. So what can they learn next? So if you feel that the introduction to bioinformatics program is something that you already know, what are these different things, and you want more advanced courses, so we have two upcoming courses and we'll have a few more. So this is the data science program, which is also starting on 12th October. So this program will be totally on the different biomedical data and their applications. And we will be using primarily the Code OmicsLogic platform for this. So this is a data science program, which is an advanced program, which you can consider joining. And this is specifically related to biomedical data analysis using R and Python. So you will be able to go through that in this program. And in December, we have the transcriptomics program, which we are repeating again this year. So this is the RNA-seq analysis program, which you can again check. So I'm going to be adding both the program links right here on the chats. And if you have any questions, please let us know. I'm open to taking questions right now. So thank you for joining today. And any questions you have or any problems you have in regarding the resources, you can always email me at marketing@pine.bio for more details. And we will be sharing this video with you, and all of these resources that were mentioned here, we would be emailing you the details of these resources. So if anybody has any specific requirements or you are not sure, what you want to be doing, you can email us and we will be able to guide you regarding anything related to what we were talking about today. So Dr. Mohit, over to you.
Thank you, Deeksha. Am I audible?
Yes, Dr. Mohit, you're audible.
Okay, so thank you everyone for joining today's session, and I would like to invite everyone to go to some of these resources and check the free resources and about the program. And as Victor said, that we will be available. You can reach out to us on LinkedIn and Facebook, and also through these several platforms that we discussed today. So, and if you have any questions right now, you can actually unmute yourself and ask us as well. Okay, then. If there are no more questions, I guess we will see you again next time, and we'll be looking forward for your participation in the program, and wish you. Okay. So there's a question from Abhinav. How frequently you conduct these types of courses? So Abhinav, we conduct these types of courses very frequently. So as you saw, as Disha told you that we are going to do a data science program, which is more focused on the technologies like R and Python and its application. As you know, these fields are very diverse, and quite a bit of research has already have happened. So a dedicated program helps us to have it's for a very specialized kind of an audience. So transcriptomics is another such program where we will be learning about RNA-seq data type. And similarly, genomics is there. Similarly, metagenomics is there, which will be again happening in November. So we have an entire schedule of programs that you can check out. And if you're interested, we'll
Yes, if you're interested, Abhinav, we can share these programs with you. So let us know. You can email me marketing@pine.bio for these kinds of requests. And also regarding the server platform, when somebody had asked us that if the server platform is available to students for free, it is not available for free, but during the programs, it is available at no additional cost. So anybody who is also doing their own research and requires like a research license or is thinking about something on that aspect can also reach out to us regarding these specific requirements. But as a whole, the Introduction to Bioinformatics program is meant for students who are just starting to learn about NGS and are getting familiar with these technologies. So this is a basic program that we are offering to everyone. The others are more advanced programs, and as I mentioned again, that we will be sharing these lists. So let us know. Any more questions? Okay. If there are no more questions, then I think we will be ending today's session. And also we are going a bit over time. So thank you everyone for joining today. Reach out to us for any questions that you might have. And we look forward to seeing you at this session. Thank you. Yeah, thank you, Lakshmi.
Yeah, thank you everyone. I see Norman is joining right now, but just to tell that the session has just ended, and we will be sending you the recording of this session. So anyone who have missed the session will get an opportunity to review the session again. So thank you again, and bye bye.
Okay, thank you. Thank you.
Thank you.